Welcome to foodfeedinfo.com. This is part 4 of Carbohydrates Metabolism. In this lecture, we will cover glycogenolysis and glycogenesis. I would like to acknowledge the books which I have used during the preparation of this lecture and slides. The first one is Principles of Biochemistry. This is the 6th edition of Lehninger. And the second one is Animal Nutrition by McDonald, 7th edition. What is glycogen? Glycogen is a reserve polysaccharide, and animals store excess glucose by polymerizing it to glycogen. Glycogen is also known as animal starches, and it occurs in liver, muscle and other animal tissues. It is about 10% of the liver weight and 1 to 2% of the muscle weight. If we look at the structure of the glycogen, it is similar to that of amylopectin. However, the difference between glycogen and amylopectin is the branching. In case of glycogen, the branches are shorter and more frequent. The molecular weight of glycogen vary considerably according to the animal species, the type of tissue and the physiological state of the animal. Glycogen is the main carbohydrate storage product in the animal body, and it plays an essential role in energy metabolism. This is the structure of glycogen. If you look at this structure, so it is similar to that of the amylopectin structure. There is a straight chain of alpha 1,4 glycosidic bond, and there is a branching at alpha 1,6 glucosidic linkages. What is glycogenolysis? It is defined as breakdown of glycogen to glucose 6-phosphate. Glycogen is present in liver, muscle and other animal tissue, and when body needs energy or there is a deficiency of glucose in the body, then glycogen get hydrolyzed and that is then converted into glucose. Here is the examples, like during exercise and stress, there is a requirement of more glucose. So in that situation, this glycogen get hydrolyzed to provide glucose to the to the cells. Here are some facts about glycogenolysis. Like in case of muscle, the muscle glycogen is a quick source of energy for both aerobic or anaerobic conditions, and during exercise or when muscles they are highly active, this glycogen can be exhausted in less than an hour during that vigorous activity. When we talk about liver glycogen, it serves as a reservoir of glucose for tissues like neuron, which are unable or which cannot use fat as fuel. So it provides energy when dietary glucose is not available, particularly during meals or during a fast. Liver glycogen can be depleted in 12 to 24 hours. Just to give you an idea about breakdown of glycogen in gastrointestinal tract. So in the GIT, the amylases cause the breakdown of starch into dextrin, then into maltose and glucose. So in the same way, the glycogen is converted or hydrolyzed into dextrin, then maltose and then into glucose. So dextrin is an intermediate product of the hydrolysis of starch and glycogen. It is then converted into maltose and then into glucose. Glycogenolysis is the breakdown of glycogen, and it occurs in liver when blood glucose level decreases. It also occurs in muscle during its contraction or muscular exercise. The cellular site where glycogenolysis is going on, that's a cytosol, And the most important thing to be keep in mind, it is not a reversal of glycogenesis. It means the breakdown of glycogen is not the reversal of synthesis of glycogen. Now about the steps of breakdown of glycogen. During the first step, there is a removal of a terminal glucose residue from the non-reducing end of a glycogen chain. This happened by the action of inorganic phosphate and glycogen phosphorylase. The second step is the debranching of glycogen residue. Now about the enzymes which are required during glycogenolysis. The first one is glycogen phosphorylase, and the second one is glycogen debranching enzyme, and the third one is phosphoglucomutase. So these are the three enzymes which are required. In skeletal muscle and liver, the glucose unit of the outer branches of glycogen enter the glycolytic pathway through the action of these three enzymes. I would like to acknowledge the Lehninger, who is the author of Principle of Biochemistry 6th edition. I have taken these breakdown of glycogen pictures or structure from that book. Starting with the first step, if you look at the picture on the left side of the picture, there is a non-reducing end of glycogen, and due to the action of glycogen phosphorylase, it catalyzes the reaction in which alpha-1,4 glycosidic linkage between two glucose residues at a non-reducing end of glycogen undergo attack by inorganic phosphate. It causes removing the terminal glucose residue as glucose 1-phosphate. If you look at the bottom of the picture and on your left side, there is a glucose 1-phosphate formation due to glycogen phosphorylase. The Lehninger explained this. This phosphorylases reaction is different from the hydrolysis of glycosidic bond by amylase during intestinal degradation of dietary glycogen and starch. In phosphorylasis, some of the energy of the glycosidic bond is preserved in the formation of phosphate ester like glucose 1-phosphate. Again, just to explain in detail, so look at the non-reducing end of the glycogen. Due to glycogen phosphorylase, there is a stepwise separation of glucose molecules that result in the production of glucose 1-phosphate molecule. So this process continues until it reaches the fourth glucose unit from a branch point. So if you look at this on the right side of the picture with the red arrow, so there are 4 glucose molecules where the branching start. So it means the rest of the glucose molecules, they repeatedly get separated by glycogen phosphorylase, and there is a production of glucose 1-phosphate molecule. Now look at this picture. The upper portion of the picture shows the complete glycogen structure, and due to the activity of glycogen phosphorylase, there is a production of glucose 1-phosphate molecules. But this glycogen phosphorylase, it acts repeatedly on the non-reducing end of the glycogen branches until it reaches a point 4 glucose residue away from an alpha-1,6 branching point where its action stops. So if you look at the right side and the bottom of the picture, there is a branch structure with 4 glucose molecule at alpha-1,6 linkage. So to continue the degradation process, this further degradation by glycogen phosphorylase can occur only after the debranching enzyme, which was known as oligo alpha 1,6 to alpha 1,4 glucan transferase. So it catalyzes two successive reactions that transfer branches. Once these branches are transferred and the glucosyl residue at carbon number 6 is hydrolyzed, then glycogen phosphorylase activity can continue. Glucose 1-phosphate can enter glycolysis or in liver it replenish the blood glucose. This is what I have discussed in the previous slide. There is a debranching enzyme which was formerly known as oligo 1,6 alpha 1,6 to alpha 1,6 glucan transferase. It catalyzes two successive reactions that transfer branch glucose molecules. So if you look at this as a first step, the transferase activity of debranching enzyme result in the transfer of these yellow glucose molecules from alpha 1,6 to alpha 1,4. So you can see here in as a result of this first step or debranching enzyme activity, the yellow glucose molecules, they get attached with the straight chain while leaving the one glucose molecule as a branch chain structure alpha 1,6. So as a next step, what happened? The alpha 1,6 glucosidase activity of debranching enzyme occurs. So it causes the separation of this glucose molecule from the branch, and it releases this unbranched alpha 1,4 polymer. So this is a substrate for further phosphorylase reaction. So as a result of glycogen phosphorylase, there is a production of glucose 1-phosphate molecules. So we can say glucose 1-phosphate is the end product of glycogen phosphorylase reaction. This glucose 1-phosphate it can enter glycolysis or in liver it replenishes the blood glucose. This glucose 1-phosphate is then converted into glucose 6-phosphate by phosphoglucomutase. This phosphoglucomutase it catalyzes the reversible reaction and result in the production of glucose 6-phosphate. This glucose 6-phosphate which is formed as a result of glycogenolysis, it can be used for the production of glucose or energy. In case of skeletal muscle, it can enter glycolysis for the production of energy, particularly when muscles they are contracting and they need energy, and it can also hydrolyze or utilize through hexose monophosphate shunt. Whereas in case of liver, the glycogen breakdown serve or utilized for a different purpose. It is used to release glucose into the blood when blood glucose level drops, as it does particularly between the meals. So this step requires an enzyme which is glucose 6-phosphatase. So that is present in the liver, and that result in the production of it is present in the liver and kidney but not in the other tissues. The reason being the muscle and adipose tissues lack glucose 6-phosphatase activity, so they cannot convert the glucose 6-phosphate formed from glycogen breakdown to glucose, and these tissues therefore do not contribute glucose to the blood. From this complete slide, we can say the glucose 1-phosphate molecule produced as a result of glycogenolysis is converted into glucose 6-phosphate through phosphoglucomutase, and this glucose 6-phosphate within the muscle it oxidize and produce energy through glycolysis or hexose monophosphate shunt during muscle contraction. And in the skeletal muscles there is no production of glucose, the reason being the skeletal muscles, they lack glucose 6-phosphatase. However, in the liver there is a release of glucose to the blood to maintain the blood glucose level, the reason being there is a presence of glucose 6. So here is one question: why energy production from glycogen is slightly more efficient than it is from glucose? So I hope you would be able to answer this question, why energy production from glycogen is slightly more efficient than it is from glucose. Next is glycogenesis, that is the synthesis of glucose. Synthesis of glucose occur in liver and muscle when there is a plenty of glucose. So when glucose is present in excess amount, so it is converted into glycogen. The cytosolic pathway are requiring ATP and UDP glucose for the synthesis of glycogen. The first step in the synthesis of glycogen or glycogenesis is the synthesis of uridine diphosphate glucose, or simply we can say UDP glucose. So there are 3 steps which are involved in glycogen synthesis. The first is the activation of glucose. So this activation of glucose is brought by the formation of UDP glucose. UDP glucose is the activative form of glucose for glycogen synthesis and also for the synthesis of other complex carbohydrates. So what happened during this UDP glucose synthesis? The glucose is first converted into glucose 6-phosphate. That is similar to the step that we have in case of glycolysis through this hexokinase or glucokinase activity, and with the utilization of 1 ATP there is a formation of glucose 6-phosphate. So in the next step, through phosphoglucomutase, glucose 6-phosphate is converted into glucose 1-phosphate. So in the next step there is a utilization of UTP and production of 2 inorganic phosphorus. Glucose 1-phosphate is converted into uridine diphosphate glucose by the action of UDP glucose pyrophosphorylase enzyme. So in this way the active form of glucose or activated glucose in the form of UDP glucose is synthesized. The next step in glycogen synthesis is the synthesis of linear chain of glucose. This step requires glycogen primer, which is a protein called glycogenin with 3 to 4 glucose attached to it. So if you look at this figure, the arrow indicates the glycogen primer, where you can see the glucose molecules linked together through alpha 1 for glycosidic bond. With the action of glycogen synthase enzyme, the UDP glucose molecule get attached with the glycogen, that result in the formation of a linear chain of glucose molecules. They are linked together through alpha 1 for glycosidic bond, and as a action of this glycogen synthesis, we can say there is a synthesis of linear chain of glycogen and there is a production of 1 UDP molecule. In the next step there is a action of branching enzyme. If you just remember the glycogenolysis, there is a debranching enzyme. The debranching enzyme actually cause the removal of the glucose molecule from the branch, whereas during this glycogenesis this branching enzyme result in the formation of branches. Just to indicate the one thing, the glycogen synthase it repeatedly attach glucose residue from UDP glucose and creating the alpha 1,4 link. So it is done from non-reducing end of the molecule. So during this third step, which is brought by the action of branching enzyme, it transfer a linear chain of 6 to 8 glucose residue to another chain of glucose, result in the formation of alpha 1,6 linkages. Thus branching takes place. To the newly formed branch, glycogen synthase again add glucose unit by alpha 1,4 linkages with further branching and elongation to form the glycogen. So in this way the branching occurs and there is a formation of branch structure of the glycogen. Next is the regulation of glycogen metabolism. Glycogen synthesis and glycogen degradation is achieved by phosphorylation of the key enzymes glycogen synthase and glycogen phosphorylase. Glycogen synthase is active in the dephosphorylated state, whereas glycogen phosphorylase is active in phosphorylated state. So glycogen synthesis and glycogen degradation, they are not active at the same time, because the synthase is active in dephosphorylated state, whereas the phosphorylase is active in the phosphorylated state. So that's the reason that both are not active at the same time. Little bit about the hormonal regulation of this glycogen metabolism. So enzymes are phospho or dephosphorylated and they are controlled by the hormone. Hormones here are the glucagon. It stimulates glycogen degradation in the liver during the fasting state. Second one is epinephrine and norepinephrine, which are known as catecholamines. They stimulate glycogen breakdown in liver and muscle during physical activity. And insulin, it stimulates the glycogen synthesis in the liver and skeletal muscle. Because insulin, we all know, okay, it is produced when there is a rise in blood glucose level, so it causes the utilization or metabolism of the glucose. So it stimulates glycogen synthesis in liver and skeletal muscles. And the enzyme or metabolic control of glycogen metabolism. Glucose 6-phosphate is an inhibitor of glycogen phosphorylase in the muscle. The glycogen synthesis is favored when substrate is available. Then AMP, adenosine monophosphate, is an allosteric activator of glycogen phosphorylase in muscle. So the glucose is an inhibitor of glycogen phosphorylase in the liver. So this is how enzymes, and there is a metabolic control of this glycogen regulation. Again one question: why anaerobic glycolysis from glycogen produces more ATPs than glucose? Why anaerobic glycolysis from glycogen produces more ATPs than glucose? So looking forward to have answer in the comment box. Thank you for watching, and I hope you learned a lot during this lecture, and looking for your answers. Thank you again.